everyone to today's class of genetic code and organization of mammalian genome under the subject Biochemistry BP 203T. Today we are going to study about genetic codes and how these genetic codes help us in synthesis of DNA and RNA. So let us discuss a little bit an introduction of genetic codes. The 3 nucleotide, that means the triplet base sequence in mRNA that act as code word for amino acid in protein constitute of genetic codes or the simply codons. And what are those codes? Like if we see the dictionary of nucleotide bases like A, G, C and U. Why we are saying U here? Because we are talking about the proteins, that means the synthesis of proteins from RNA. And RNA do not have thymine in it. That's why there is an incorporation of uracil, because RNA has uracil in it, and then finally that is incorporated for the synthesis of proteins. Now, the codons are composed of 4 nucleotide bases, namely purines, that is adenine and guanine, pyrimidines, that is cytosine and uracil. These 4 base produces 64 different combinations of 3 base codons. The nucleotide sequence of codon on mRNA is written from the 5' to 3' end. 61 codons out of 64 codons, 61 codons code for 20 amino acid, and the 3 codons, that is UAA, UAG and UCA, that do not code for amino acids. They act as a stop signal in protein synthesis. These 3 codons are collectively known as termination codons or nonsense codons. So, out of 64 combinations, out of 64 different combinations, only 61 are the which are code for 20 amino acids, and rest 3 are the nonsense codons which do not code for anything. They are called as amber, ochre and opal codons. Next, the codons AUG and sometimes GUG are the chain initiating codons. These are the chain, that means when these codons are present, that means there is an initiation of that coding happening. So, what are the characteristic of this genetic code? First characteristic is the universality. That means there are so many codons and only 20 amino acids are there. That means the same codons are used for code of same amino acid in all the living organisms. Hence, genetic code is appropriately regarded as universal. There are, however, a few exceptions, like AUA is the codon for methionine mitochondria, and the same codon AUA codes for isoleucine in the cytoplasm, with some exceptions noted. The genetic code is universal. So, there are some exceptions always there, but rather more exceptions there are universality we can see in the genetic code. Second is our specificity. A particular codon always codes for the same amino acid. Hence, the genetic code is highly specific or unambiguous. That is, like see the example, UGG is the code for tryptophan. Okay, so there is always a particular codon for the same amino acids. Next is non-overlapping. That means how do we read these codes? The genetic code is read from a fixed point as a continuous base sequence. It is non-overlapping, it is commaless and without any punctuation. For instance, let us see the example. If there is a sequence of UUU, CUU, ACA, CGA, like this is the sequence. So, how we are going to read it out? We are going to read it out in 3 pairs, 3 codon per pair we can read it out. Like we read it out like UUU, CUU, ACA, GCG, like that, or CGC, whatever the sequence will be. So, like that we going to read it out in the form of 3,3. Next, degeneration. Most of the amino acids have more than one codon. The codon is generate or reductant. Since there are 61 codons available to code for only 20 amino acids. For instance, glycine has 4 codons. The codons that designate the same amino acid are called as synonyms. Most of the synonyms differ only in the third, that is 3' end of the codon. So, most of the amino acid that has only one codon, that is the code is degenerate or reductant, that can happen, okay, because there are 20 amino acids. So, that is a possibility, there is a chance of the degeneration. So, codon-anticodon recognition, that how will they recognize themselves? We know the general rule that A, that is adenine, will pair with uracil, guanine will pair with cytosine. So, adenine double bonded with uracil, whereas guanine is triple bonded with cytosine. So, the codon of mRNA is recognized by the anticodon of tRNA. They pair with each other in anti-parallel direction, 5' to 3' of mRNA with 3' to 5' of tRNA, because I told you that they are the two strands of DNA, RNA, proteins, whatever happening here always happens in complementary strands. That means, if one strand runs from 5' to 3', the other will definitely runs from 3' to 5'. The usual conventional complementary base pairing, A double bonded U, whereas G triple bonded C. Here I have written G double bonded. This is just to show you that these two are bonding together. This is not the bonding style. This is not the double bond or triple bond. This is like A is equal to U and G is equal to C. Occurs between the first two bases of codon and the last two bases of anticodon. The third base of the codon is rather lenient or flexible with regard to the complementary base. The anticodon region of tRNA consists of 7 nucleotide and it recognizes the 3-letter codon M in mRNA. Okay. Now, to understand the fashion how they are bonded, let us discuss the Wobble hypothesis. Now, what is Wobble hypothesis? Wobble hypothesis was given by Crick. We have known the name Watson and Crick from them. Crick has told us the Wobble hypothesis. In this phenomenon, a single tRNA can recognize more than one codon. He said that. This is due to the fact that the third base, that is 3' base in the codon, often fails to recognize the specific complementary base in the anticodon, that is 5' base. This is a normal thing, that is the single tRNA can recognize more than one codon, because this is a fact due to the third base, 3' base in the codon often fails to recognize the specific complementary base in the anticodon 5' base. Wobbling is attributed to the difference in the spatial arrangement to the 5' end of the anticodon. The possible pairing of 5' end base of anticodon of tRNA, of course, with the 3' end base of codon mRNA is given. So, like that, if the anticodon is C, the codon will be G. If the anticodon is A, the codon will be U. If the anticodon is U, it will be G or A. For the non, like let us see. Let us see. This is the conventional base pairing. C is bonded with G, A is bonded with U. But in the non-conventional base pairing, it is given that along with the conventional base pairing, there is a non-conventional base pairing, that is U will be bonded with G and G will be U. This is the non-conventional base pairing. Now, the Wobble hypothesis that explains the degeneracy of the genetic code, that is the existence of multiple codons for single amino acid. So, you can see there are 61 codons. Here are, this whole cycle shows us the 61 codons for amino acids. The number of tRNA is far less, that is around 40, without which is due to the wobbling. This is all due to the wobbling. There are actually all the 64 codons present, but these 3 codons are stop codons, stop signaling codons. So, rest of the codons are 61, which we use for coding for amino acids. So, here are 60 codons present. Next, organization of eukaryotic DNA. Now, let us see, that was all about our genetic codes. What are genetic codes? How do they code for our proteins, for our RNAs? We have discussed. Now, we are discussing about the organization of eukaryotic DNA. As we all know, eukaryotic organisms are different than prokaryotic organisms. Prokaryotic organisms are also called as simple celled organisms, whereas eukaryotics are complex cell organisms. That means they have some processes more complicated than prokaryotic. So, in this we are going to study about the organization of the eukaryotic DNA. So, in the eukaryotic cell, the DNA is associated with various proteins to form chromatin, which then gets organized into into chromatin, which then gets organized into compact structure called as chromosomes. So, here we are going to study about chromatins, the chromosomes, how they are packed, what are the types of it, and so on. So, how the packing of DNA into a chromosome happens. A somatic human cell, it has 46 chromosomes. Generally, we call it 23 pairs of chromosomes, then it is 46 chromosomes with a total length of DNA which is equal to 1 to 2 meter. But the size of nucleon is billion or million of times smaller, that is 6 into 9 micrometer. Such an enormous amount of DNA, that is 2 nanometer wide, must be packed by a elaborate procedure into the nucleus and then in chromosomes of the dividing nuclei with precision. DNA is associated with a group of basic proteins which are called as histone proteins, which are very important in the structure of DNA. Histone proteins to form a nucleoprotein complex having about 50% DNA and 50% protein, which are called as chromatin. So, histone proteins forms a nucleoprotein complex in which there is 50% of DNA and 50% of protein, which is completely called as chromatin. So, chromatin stains with basic dyes. How do we estimate what chromatin is? It has some properties like chromatin stains with basic dyes like hematoxylin and fishin. It occurs in two forms in the resting non-dividing nucleus. First is euchromatin with loose structure and is lightly stained, that is negatively heteropyknotic, and the other one is heterochromatin that are more condensed and deeply stained. They are called as positive heteropyknotic. Only euchromatin is generally active, but in dividing cell the chromatin condenses and assembles into specific number of well defined chromosomes for the proper distribution of replicated DNA among the daughter nuclei or cells. So, the first we are going to study about now the types of proteins present in here. First, there are histone proteins. There are 5 types of histones: 1H1, 2H2A, 2H2B, 2H3 and 2H4. They are rich in arginine and histidine. Both are amino acids that provide them a positive charge which forms ionic bond with negative charge on the phosphate group of DNA. The amino acid sequence of eukaryotic histones are highly conserved in different groups with little variation. However, H1 histone shows variation in structure in different species and even in different tissues of the same organisms. Now, second is nucleosome. First was histone protein, now we are studying about nucleosome. Two molecules each of H2A and H2B, H3 and H4 histones form an octamer complex called as nucleosome core, around which the DNA wraps two turns, that is around 200 base pairs in a left-handed orientation. DNA enters and leaves the nucleosome at the same vicinity where one molecule of H1 histone may seal it. Virtually all of DNA is associated with nucleosomes. During the interphase, the chromatin appears as bead on string structure with nucleosome connected by an intervening stretch of DNA of a variable length of 50 to 60 base pairs. It is called as linker DNA and is associated with the H1 histone. DNA form 1.65 nanometer, that is around 146 base pairs around each nucleosome, 10 nanometer in diameter. 10 nanometer in diameter, which is called as the core DNA and resistant to digestion by the nuclease. Now, third, that is, this was all about the nucleosome. Third is hierarchy of packing of DNA. Now, how the DNA is packed. There are several levels of organization that results in tight packaging of DNA into the chromosomes. First, that is in that first wrapping around the nucleosome. In the first step, extended linker DNA makes two complete turns around each nucleosome, that is around 200 base pairs, two folds. It go for two complete turns and is sealed by H1 molecule. It results in 7-fold compaction of DNA. The nucleosome come closer to form a 10 nm thick fiber. That's how the proteins, the DNA are folding together, wrapping together. Next, the formation of 30 nanometer quick fiber or chromosomes. The 10 nanometer fiber in condensed further like a spring to form a 30 nanometer fiber. There are 6 nucleosomes per turn which are organized into a solenoid. Histone H1 is required for formation of the 30 nanometer fiber. The packaging ratio of DNA at the level is about 40 folds. Now, the formation of metaphase chromosome from 30 nanometer fiber. The 30 nanometer fiber or chromonema forms loops on the central non-histone protein scaffold of the chromosomes. Average length of loop in the looped domain is about 50,000 to 1 lakh base pair of DNA. The packaging ratio of the looped domain is about 750 fold, which is the typical euchromatin in heterochromatin and dividing chromosomes. The looped domain are further condensed to give a still higher packing ratio. At metaphase, a typical human chromosome measures about 4 to 5 micrometer in length that contains about 75 nanometer of DNA, which fully extended. The packing ratio falls in the range of 15,000 to 20,000. This is the organization chart of our DNA. So, you can see there are naked DNA double helix structure. This is the basic structure which we study about. the length of this is around 2 nanometer. Then there is a beads on string forms of chromatin, there is a incorporation forms of chromatin in this part of DNA. So, these are the chromatin here. Now, so the length becomes 10 nanometer. Then 30 nanometer chromatin fiber composed of nucleosome. Now, you can see there are chromatin, there are nucleosomes present in here. So, the structure becomes 30 nanometer, it has increased. Then the chromosome in an extended form. Now, the chromosomes are in the extended form, the length becomes 300 nanometer. Now, again the structure, the wrapping of the DNA happens, and here condensed form of chromosome, you can see the condensation of the chromosomes, it becomes 700 nanometer, and finally when there is a formation of metaphase chromosome, a proper structure of chromosome, then it is 1400 nanometer. So, starting from 2 nanometer to 1400 nanometer, we have seen various phases of the formation of the chromosome structure, the DNA structure. These are the references I have used. Now, let us revise once again about this topic, that what are genetic materials, genetic codes, and finally the organization. So, a quick revise about genetic codes are 4 codes we all know when we make RNA to protein, and then after protein we use that codes in the form of amino acids. So, there are 4 codes A, G, C and U, adenine, guanine, cytosine and uracil, which codes for 64 different combination for codons. So, out of 64 different combination codons, 61 codons are the codons which codes for 20 amino acids, rest 3 codons, that is UAA, UAG and UCA, are called as stop codon, nonsense codon, termination codon, known as amber, ochre and opal. These are the stop codons. Now, what are the characteristics? We have seen genetic codes are universal, they are very specific, they are non-overlapping, they are degenerate with some changes, and we have already studied about codon-anticodon recognition, how they will read one another. If there is a incorporation of adenine, it will definitely bond with the uracil. If there is a presence of guanine, it will definitely bond with cytosine, and vice versa. Now, the Wobble hypothesis, which was given by Crick, here we have seen the single tRNA can recognize more than one codon. He suggested that. So, there is a conventional base pairing method and the non-conventional one. The non-conventional one, he has says that uracil will bond with guanine and guanine with uracil as well. These are the Wobble hypothesis, in which there are 64 codes are there. 3 codes are nonsense codes, and rest 61 are coding for 20 amino acids. This we have studied organization of eukaryotic DNA, how DNA is formed. We have studied here about chromatin. In chromatin we have studied about euchromatin, we have studied about heterochromatin, we have studied histone proteins. There are 5 types of histone proteins. The use of those histone proteins, we are using nucleosome. We have studied about hierarchy of packaging of DNAs. We have we have studied about the formation of 30 nanometer fibers. Then after 300 nanometer fibers, likewise we have seen the full structure from how a 2 nanometer DNA diameter, which will come upon when there is a full formation of metaphase chromosome of 1400 nanometer, how it looks and how it is happening. These are the references. Thank you once again. Hope you enjoyed the class.